Hey guys, welcome to another A-level biology video, and this time we will be talking about the topic genetic technology. And firstly, let's have a background of this topic. So we know that by the 1950s and the 1960s, the structure of DNA and the way in which it codes for protein synthesis was worked out. And since then, the knowledge that we got has developed so much that we are now capable of actually changing the DNA in a cell and thereby changing the proteins that the cell can synthesize. Cool, right? And that isn't only it. We can also sequence the nucleotides in DNA and compare the nucleotide sequences in different organisms. And what that means is that we can figure out the nucleotide sequence in a particular gene in a particular organism. So we know the order of the nucleotides. And if we find the order of the nucleotides in the same gene or a similar gene in other organisms, we can basically compare the orders, right? And in addition to this, we're also now capable of carrying out genetic tests to see if people are carriers of certain genetic diseases. And what that means is that if we can test the genome of a perfectly healthy person to see if he or she carries a recessive allele, which while in the person is not in a homozygote formation, but were it to be in a homozygote formation in any offspring, that offspring would have a genetic disorder. So people can test for sickle cell anemia allele, right? They can do that sort of thing, especially if the other person, the person they're marrying, is a confirmed carrier of sickle cell anemia. Then it is a good idea that the other person also have a genetic test, because if the other person turns out to be a carrier as well, although it's a bit rare because it isn't all that common, then chances are that any offspring they have will have sickle cell anemia, which is a really bad genetic disorder, and the chances are 25%. That is 1 by 4 chance that the first offspring that they will have will have sickle cell anemia, right? And in a few cases, but not in all of them, and I will explain why later, after those genetic tests, we can actually treat people who have genetic disorders. You'd be surprised, but it is a possibility in some genetic disorders. So we can summarize that gene technology has brought huge benefits to many people, but there are consequences that we may not yet be able to foresee. And these technologies raise social, economic, and ethical issues that we must confront. And this is actually an overview of all the topic, this entire topic. So genetic engineering, and what is the aim or the basic principle of gene technology? It is the removal of a gene or several genes from one organism and the transfer into another. And why do we do that? It is so that the gene that we have transferred, which was not previously present in the host organism, is now able to be expressed within the host organism. And this could be for a number of different reasons. We could be doing this for a number of different reasons. We do this in gene therapy. Alright, we can do this in gene therapy, and we do this for commercial purposes. Because in gene therapy, for example, a patient of cystic fibrosis at a particular gene locus has two copies of an identical recessive allele, which makes a non-functional CFTR transporter protein, and because of that, that person has abnormally thick mucus and therefore has difficulty breathing. And now this issue, this genetic disorder, can be rectified if the person were to somehow gain a dominant version of the allele for that gene locus, right? So basically, with the help of a vector, a dominant allele is somehow transferred into that patient's genome, or at least into the genome of a few cells, right? It's a bit difficult to permanently incorporate it into the genome of all the cells, especially for an adult patient. As of yet, it is impossible, but it is possible that it can be incorporated into the genome of quite a few of the relevant cells. And now what happens is that this gene, or more accurately this allele, which was not normally present or naturally present in the genome, has now been introduced via genetic technology. And now this dominant allele actually suppresses recessive allele and codes for the functional CFTR protein, because now this gene which has been introduced, this allele which has been introduced along with a promoter, can be transcribed and translated by the enzymes that are present in the cell of that organism. So this is an example. In genetic therapy, in commercial aspects, we produce insulin in this manner. How do we do that? We basically know that insulin is not produced in bacteria because they do not have any reason for that. You'd know bacteria most certainly do not have glycogen deposits, right? You know this. Is isolated from the genome of a normal person, right? And after isolation and removal, this gene is somehow transferred into the genome of a bacteria, and the bacteria is now cultured in large amounts. And because bacteria reproduce asexually, all of the offspring will have that insulin gene and would be able to produce the insulin in certain conditions, when given certain conditions, and the insulin that they produce can then be harnessed. So the basic principle of genetic technology is the ability to remove a gene from the genome of one organism, transfer it to the genome of another which does not normally contain the gene or the allele, and then somehow ensuring that the host is able to successfully express the gene product. And the DNA that has been altered by this process and which now contains length of nucleotides from two different organisms is called recombinant DNA, and it is actually mentioned in the learning outcomes that you need to be able to define recombinant DNA. So in this scenario, it is the DNA of the E. coli, to which the insulin gene has been transferred, which will be termed as recombinant DNA. So formally we may define recombinant DNA as DNA that contains length of nucleotides from two different organisms. And I often suggest that you actually write down these definitions and these key points when you are learning, because you often forget otherwise. So recombinant DNA is DNA that contains lengths of nucleotide, and you may underline this, from two different organisms, from two or more different organisms actually, or more different organisms, and the organisms could be of the same or of different species. They just don't have to be the same, they just don't have to be from the same individual. And this is important, it just doesn't have to have the gene, but also has to be capable of expressing it. And now expresses the gene slash genes, because it could be more than one. So successful transfer implies that the transferred genes have to be successfully expressed to produce the needed gene products. And these GMO or these transgenic organisms are used for multiple purposes, and it is part of the learning outcomes to realize the role of GMOs in food production and the social and ethical implications that follow, because so many things can be termed as GMOs. For example, there are a lot of genetically modified crop plants. Some of them have herbicide resistance, and some of them are resistant to certain insect pests, and all of these are genetically modified because by themselves naturally they do not have the genes that make them resistant to a particular herbicide or that make them resistant to certain insects. And there are social and ethical implications because there are certain risks that are foreseen, and we aren't all that sure just how credible those risks are. Maybe we're just not foreseeing the actual consequences. So recombinant DNA is made by joining pieces from two or more different sources and provides a way of overcoming barriers to gene transfer between species. So this gene transfer just doesn't have to be between the same species, can be, but can be between species as well, and we can even transfer them in different kingdoms. For example, a bacterial gene can be inserted into a plant, or a human gene can be inserted into a bacterium. And an example for bacterial gene being inserted into a plant is for herbicide resistance, because there is a herbicide, I believe it's called glyphosate, and it's usually used to destroy a lot of weeds. But if we were to use it, the crops that we want cultivated would be harmful or would be adversely affected as well. So genetically, certain genes are transferred from the bacterium into the plant that make them resistant to glyphosate. Now, how is this resistance given? Well, the thing is that organisms, as well as those plants, they produce a certain enzyme which is needed for the production of amino acids. As you well know, plants produce their amino acids themselves, and the amino acids that are produced are phenyl, alanine, and I believe tryptophan. I'm not really sure about the name. So there are three amino acids which it would not be able to produce anymore, because the glyphosate is actually an inhibitor for that enzyme. But the gene which we are transferring from the bacterium actually codes for the same enzyme that is the same purpose but is slightly different structurally, and because of that structural differences it cannot be inhibited by the glyphosate, and so the organism, the plant, effectively becomes resistant. Unlike selective breeding, where whole sets of genes are involved, genetic engineering often results in the transfer of a single gene. So they may ask you a question, what differences in selective breeding and genetic engineering? But the differences are clear enough. In selective breeding, you're just playing with a given gene pool, right? You aren't introducing new things into the gene pool. If you have a vast gene pool, and you're selecting for which components of the gene pool you want in your organisms, right? Whereas in genetic engineering, you're capable of introducing a completely new allele or a completely new gene from some other species. So there is a lot of difference there. So there are some general principles involved in genetic engineering and some techniques, and they actually have a learning outcome for this, an understanding of the tools and techniques involved. So I'd say you need to pay a lot of attention to what I'm saying. So we can produce GMOs in different manners. However, there are some commonalities. Number one, you need to identify the gene that you want transferred, and you need to get it somehow. You may cut it from a chromosome. You may make it from the mRNA, which is complementary to that gene, and you can convert the mRNA back to chromosomal DNA using reverse transcriptase. And you need to know what reverse transcriptase is, because it is mentioned in the learning outcomes. And it is an enzyme that catalyzes the conversion of mRNA back to cDNA. And thirdly, you may simply synthesize it from nucleotides. Because recently there have been a lot of advancements in DNA sequencing, so we know the sequences of many important genes, and if we know the sequences, that we know the sequence of nucleotides, we can manufacture those genes via computer modeling, and we won't need any template. Number one. Number two, now that we have acquired the gene either by using restriction endonucleases to cut it from a chromosome, and restriction endonuclease are a class of enzymes and they're also mentioned in the syllabus, so we will talk about it, or by synthesizing it via nucleotides, or by converting mRNA to cDNA, whatever, we will have the genes, but we might not necessarily have a whole lot of them. We might have a few copies, but we may want to have a lot more of them so we can use it with ease, and so we use something called PCR to amplify the DNA or to make multiple identical copies of the gene. And PCR is actually mentioned in the syllabus 2 times, so it is important. And now that we have multiple copies of the gene, which we have either successfully isolated, produced, or converted, now we need to insert it into something called a vector. And a vector is something which delivers the gene to the cells of the organism which we want to be genetically modified. Examples are plasmids, viruses, and libraries. And genes that code for easily identifiable substances that can be used as markers. So these are some things we need. So restriction endonucleases, and why do we call them that? Because these enzymes are naturally found in bacteria, and in the bacteria they basically serve to restrict viral invasions, because they cut up viral DNA so that viruses cannot destroy the bacteria, and so we call them restriction endonucleases. And this isn't just one enzyme. This term actually refers to a whole class of enzymes, right? And yep, they're found in bacteria. They're capable of recognizing and breaking down the DNA of invading viruses, which we call bacteriophages. And bacteria make these enzymes to cut phage DNA into smaller pieces. And exactly how do they break up the DNA? They basically cut the sugar phosphate backbone of the DNA at specific places within the molecule. And the term specific places shouldn't really surprise you, because like all enzymes, restriction endonucleases are also really specific, meaning that one kind of restriction endonuclease will only cut up the DNA at the sugar phosphate backbone, and the place where that specific enzyme will cut up will correspond to a specific base sequence. And this is why we call them as endonucleases, because they're found within, and their role in bacteria is to restrict a viral infection, hence the name restriction endonuclease or restriction enzyme. So each restriction enzyme, and you need to learn this carefully because they've asked for restriction enzymes in the syllabus, they bind to a specific target site on DNA, bind to a specific target site on DNA, and cut at that site. So the reason I'm writing this down is because to me it's a bit difficult to explain this particular sentiment in my own words for whatever reason. So I write stuff down which I find difficult to verbalize by myself in my own words. Bacterial DNA is protected from such an attack either by chemical markers or by not having the target sites. So if you don't have the target sites, the restriction endonucleases really cannot do anything about it. And if they do have, there will be certain chemical markers which will either protect the site or signal to the enzymes that this restriction site is off limits. So they can actually ask you this question like, why don't those restriction enzymes attack their own bacterial DNA? You can write bacterial DNA is protected from restriction enzymes, and you might write from its own restriction enzymes either by chemical markers, and chemical means it will have to be some sort of a molecule like a hormone or some kind of secondary messenger, either by chemical markers or by not having the target sites. And these target sites are actually also called as restriction sites because this is a more specific term. You might want to use that when they ask you a question, because then you wouldn't have to specify what the target site means if you use restriction sites. And there are specific sequence of bases. Specific sequence of bases. So if they ask you why are restriction endonucleases considered to be specific, you can say they are considered specific because they can only bind to DNA at a certain specific target site with a particular cut the sugar phosphate backbone at that spot. Good enough for 2 marks. And you can also give an example if you remember the name of a particular restriction enzyme, but I don't think you need to remember those because nothing of the sort has been mentioned in the syllabus. For example, there is a restriction enzyme called PAM-HI, cool name, which always cuts DNA where there is a GG-ATCC sequence on one strand and its complementary sequence CCTA-GG on the other. So this is interesting. So if you were to draw a double-stranded DNA, and somewhere in the middle we have this sequence which is G, G, A, T, C, C, and right in front of it we have C, C, T, A, G, G. So if BAM-HI is produced, the only place or the only target site or restriction site it has is this GG80CC sequence with the complementary sequence right in front of it. This is literally its only restriction site. It may have many such restriction sites on the same organism, but the restriction site would be the same, right? This is actually pretty interesting, and you will notice that this sequence reads the same in both directions because it's GG80CC, GG80CC, and in this scenario it is a palindrome, but not all restriction sites are palindromic. This is also important. This is a palindromic, that is, reads the same in both directions. So this is a palindromic restriction site, but not all restriction sites are necessarily palindromic. They can be, but not all of them. Right? Restriction enzymes either cut straight across the sugar phosphate backbone, right? Or they cut in a staggered fashion. So if they cut straight across the sugar phosphate backbone, you will get blunt ends, but if they cut in a staggered fashion, you will get sticky ends. So what do we mean by this? If they were to ask you to define sticky ends, what would you say? Sticky ends are short lengths of unpaired bases. Are short, and you can underline short lengths of unpaired bases, and you can also underline unpaired because these are the key terms in this definition. They are known as sticky ends because they can easily form hydrogen bonds with complementary sequences of bases on other pieces of DNA that are cut with the same restriction enzyme. So they ask you to find sticky ends, you reply with that sticky ends are actually short lengths of unpaired bases. And they ask you, alright, so you're telling me that these are short lengths of unpaired bases. That's all good and well, but why are they called sticky ends? What would you reply then? You would reply that they are called sticky ends because they can easily form hydrogen bonds with complementary sequences. And you know the meaning of the term complementary from your study of nucleic acids. With complementary sequences of bases on other pieces of DNA, and the other is important for this definition, on other pieces of DNA cut with the same restriction enzyme. Now this is important. So what are the keywords? They can easily form hydrogen bonds with complementary sequences of bases. That is another keyword. On other pieces of DNA. That is another key term. With the same, that are cut with the same restriction enzyme, and same is another key term. So when long pieces of DNA are cut with a restriction enzyme, there will be a mixture of different lengths. Because if you have a long piece of DNA, there might be numerous restriction sites at various distances, and at each restriction site the DNA will be cleaved, and so you will get a mixture of different lengths because the restriction sites are not necessarily equally spaced. Right? And when do we need to use restriction enzymes? Previously we studied that we need these to isolate a particular fragment of DNA, and for the purpose of inserting that gene, inserting that length of DNA into the genome of some other organism. But if we were to use a restriction enzyme, chances are that the chromosome will have numerous restriction sites, and not only the ones that correspond to the length of DNA that we want. So the result is that we will get a mixture of different lengths. And how do we separate those fragments? Because we just want one particular fragment and not the whole mixture. We use something called gel electrophoresis and gene probes. So what do? We get that whole mixture and we put it in a electrophoresis machine, and so the fragments will be separated out into different bands, and each band will just contain one fragment type, right? And then you can have gene probes. You can have gene probes that are just identical or complementary to the fragment of DNA that you want. It won't be complementary with any other. So what do? Say you have 15 bands on the electrophoresis, right? So 15 different fragments, and only one of them is the one that you want. So you prepare a gene probe, and gene probe is basically a short length of base sequences which are complementary to a particular gene, right? And then you denature all the 15 bands, right? And then in turn you put those fragments onto the gene probe, right? And before putting them, you have to somehow treat those fragments, right? So that they may have, for example, some sort of a marker. They could be radioactive. You could attach some sort of a radioisotope, and then after putting them, you wash them off. And then you might somehow test for it after washing off if there is radioactivity on the gene probe. In spite of being washed off, you will know that these are the fragments you want, because only they are able to form hydrogen bonds with them. If they weren't able to form hydrogen bonds in spite of being exposed, that means they were not complementary, and so they were not the strands that you wanted. And that is important, especially the denaturation part, because if you do not denature them, then how will they form hydrogen bonds with the complementary gene probes? And if you don't mark them somehow, how will you know which adhered and which did not? So all of this is important. So let me read this again. When long pieces of DNA are cut with a restriction enzyme, there will be a mixture of different lengths. And to find the specific piece of DNA or the specific DNA fragment, you need to be able to separate the lengths of DNA using gel electrophoresis and using gene probes, and this will be described in detail later. And then you can make multiple copies of the required piece of DNA by PCR. So you have the DNA fragment that you want, and you might have like 25 copies of it, but you want say 100. You can use PCR to make identical copies of it. So explain the term sticky end. So we have 4 different lengths of DNA, A to D, and they were cut with the same restriction enzyme, and these fragments have the following number of restriction sites, and you need to state the number of fragments that will be formed from each length. So this is a bit difficult to visualize. So make up a DNA, a huge length of DNA, right? You have one, you have a second, you have a third, you have a fourth, and you have a fifth restriction site. So how many sticky ends, how many fragments will you have? You will have 1, 2, 3, 4, 5. You will have basically 6 fragments. And if we have 0, then you will just have 1 fragment. If you have 3, then what? Then you will have 1, 2, 3, 4. You will have 4 fragments. You will have the number of fragments which is 1 more than the number of restriction sites. Easy peasy. And these are 4 restriction enzymes and their target sites. So this is a palindromic restriction site, and this is from 5' to 3', and this is the complementary strand, so it's from 3' to 5'. And this is how it is cut. This, I believe, is also palindromic, and this is how it is cut. This is also palindromic and is cut in this manner, and this is also palindromic, but it isn't cut in a staggered fashion. It is cut straight through the sugar phosphate backbone, and so you will get blunt ends, whereas over here you would have gotten sticky ends. So what are blunt ends are when you cut across the sugar phosphate backbone in a straight line, so you don't get any unpaired short lengths of bases on either side of the now opened DNA. So restriction enzymes are named by an abbreviation which indicates their origin. So E. coli is the name of the restriction site enzyme, and it is called E. coli because it is derived from the bacteria E. coli. Same for BAM-HI because it is gotten from Bacillus amyloliquefaciens or something. And Roman numbers are added to distinguish different enzymes from the same source. For example, E. coli comes from E. coli and was the first to be identified from this source. Right? And this is plasmid PBR322. Now that many proteins have been sequenced, and what that means is that we know the sequence of the amino acids, it is possible to use the genetic code to synthesize DNA artificially from nucleotides rather than cutting it out of chromosomal DNA or making it by reverse transcription. So genes and even complete genomes can be made directly from DNA nucleotides without the need for a template DNA, and template DNA could be mRNA or could be a complementary DNA strand, but we no longer need it. Scientists can do this by choosing codons for the amino acid sequence that they need, right? And the sequence of nucleotides is held in a computer that directs the synthesis of short fragments of DNA. And these fragments are then joined together to make a longer sequence of nucleotides, and the sequence can be inserted into plasmids for use in genetic engineering. This method is used to generate novel genes that are used, for example, in the synthesis of vaccines. And novel genes are ones that are pretty new, and they've even been used to produce genomes of bacteria consisting of 1,000,000 base pairs. This is all very interesting. The fact that in recent years gene technology has advanced so incredibly well that we're now even capable of doing all of this without any sort of template. A go-between, right? And one type of vector is a plasmid, and plasmids have been mentioned in the syllabus, and so they're important that you get to know the properties of a plasmid that make them such good vectors, right? So these are small and circular pieces of double-stranded DNA. So small circular pieces of double-stranded DNA. So these are three things. They're small, they are circular, and they're double-stranded. And where do they occur? They occur naturally in bacteria and often contain genes for antibiotic resistance. Plus, they contain genes for antibiotic resistance. And not just one, they often contain two. And these plasmids can actually be exchanged between bacteria, even between different species. And that is why if a genetic engineer inserts a piece of DNA into a plasmid, then the plasmid can be used to take the DNA into a bacterial cell, because we are mimicking a natural process. And that is one thing you can write about if they ask you what makes a plasmid such a good vector, because it is capable of, even in its natural conditions, it is capable of transferring in between bacteria. So this makes it really ideal for transferring to a bacteria. So number one, plus naturally exchanged between bacteria regardless of species. To get the plasmids, you need to somehow get them in the first place. The bacteria containing them are treated with enzymes to break their cell wall. Alright, so we have this bacterial cell, right? And we know that bacteria have these cell walls, and they aren't really like plant cell walls because they are made of peptidoglycan and not cellulose, but they're still pretty hardy. And so the first step in getting a plasmid out is that you have to break down the cell wall. Number 1, you need to break down the cell wall, and to do that you need certain enzymes. You need to treat the bacteria with certain enzymes. So break down the cell wall via treatment with certain enzymes. Right? So we now have naked bacteria. So now what do we want to do is that we want to somehow get the plasmids or the genetic material out of the naked bacteria. And so they are spun at high speed in a centrifuge. And because centrifuge separates organelles based on density, and what that means is that the bacterial chromosomes and the smaller plasmids will be separated because they have vastly different densities. So spun at high speeds in a centrifuge. Smaller plasmids, and this is another great thing about plasmid, is because it is relatively smaller, it can be separated from the larger bacterial chromosomes via centrifuge. If they were larger, it would not have been possible to extract them so easily. Smaller plasmids separated from relatively larger bacterial chromosomes. Actually, bacteria don't really have chromosomes, it's a bigger circular DNA. Then sticky ends need to be attached to both the gene and the plasmid DNA. So the opened plasmids, because they are no longer circular because they have been cut open, and the lengths of DNA are mixed together, and some of the plasmid sticky ends will pair up with the sticky ends of the new gene. And so now they've paired up via hydrogen bonding, but their sugar phosphate backbone is still not attached. And here the third enzyme which is mentioned in the syllabus, that's the ligase enzyme, comes and does its thing. And what it does is that it links together the sugar phosphate backbones of the DNA molecules and the plasmid, so producing a closed circle of double-stranded DNA which now contains the new gene. And this is now recombinant DNA, which we just studied, right? Now, beforehand we talked about how plasmids were naturally really great at that whole acting like a vector thing, but we can also modify them to so that they're even better at their job. Because plasmids can be made artificially, for example, they can be made artificially so that they have lower molecular mass, and something that has a lower molecular mass is, as you probably already know, more easily taken up into a cell. Right? They have an origin of replication which allows for autonomous replication, meaning the plasmids replicate themselves. They have several single target sites for different restriction enzymes. Single because we just want the plasmid to be cut up at one spot when using one restriction enzyme. So several, underline several, single, you can also underline that, target sites for various restriction enzymes. In a region or in a short length of DNA called polylinker, and they might ask you for that, what's the name of the region or what is the name of the short length of DNA in the plasmid that contains several single target sites for various restriction enzymes. So remember, it's called a polylinker. Several single target sites for various restriction enzymes in a short length of DNA called a polylinker because it can link up so many different replication. So autonomous replication is a possibility, and they have a region called a polylinker, which is basically just a short length of DNA where several single target sites for different restriction enzymes are present, and they also have one or more marker genes. So plasmids all in all are really great, and this is really important, all this stuff I told you, because these have been more or less specified in the syllabus. Number one, plasmids are really great because they're small and circular. It means we can easily extract them from the bacteria, because when we centrifuge the bacteria after eliminating the cell wall via enzymatic treatment, when we spin it at high speed in a centrifuge, the plasmids can sort of be separated from the larger chromosomal DNA because there is a lot of differences in the density, a lot of differences in molecular size. So this is really great. Number 2, plasmids can naturally, because they're naturally found in bacteria, and even there they can be exchanged between bacteria of same or different species. And so we can mimic this natural process to make plasmids act as vectors. This is good. And another thing is that because of their low molecular mass, they can easily be taken up. Stuff that is a smaller size is more easily diffused. We know this. And because these plasmids have an origin of replication, so they can replicate by themselves irrespective of the cell. And because they have a region called a polylinker, which is basically a short length of DNA where several single target sites for different restriction enzymes are present, and because they have one or more marker genes, which are often antibiotic resistance genes. Two other great things is that we can make plasmids artificially